Now, I also showed you that one of the reasons, and we're going to come back to this, that when the regulatory subunit is bound to the catalytic subunit, it is inactive, is because, I'm going to just sketch that in this top board, because that regulatory subunit has inserted into the catalytic cleft of the catalytic subunit a pseudo-substrate. Pseudo-substrate. Pseudo-substrate means it looks like a substrate, but it can't be phosphorylated. And that's shown in red in the handout that you have here. Now, I want to talk a bit more about what protein kinase A actually looks like. And to do that, I'm going to show you this. So I told you before that sometimes called cyclic ATP-dependent protein kinase was the first protein kinase conceptually solved. It was the first time, it was in 1991 by a name It was the first time we actually got a look at the guts of how one of these protein kinases works. And I'd like you to notice in the front, or maybe I'll show it to you in purple, it has two lobes. Right? It kind of looks quite a bit like a kidney when you draw it this way.
Sorry, the angle here is better.
Looks a bit like a kidney. It's got what we call the M lobe for the N-terminal lobe. That's the N-terminal part of the kinase domain. And it's got this C-terminal lobe, and they have different functions. So this top lobe, this N lobe, primarily binds ATP. The C-terminal lobe primarily binds the substrates. But the catalysis happens from both residues that are contributed by the M lobe and the C lobe, or the top lobe and the bottom. Okay, and then I've given you, let's see if I can show this to you. And you can see here, and I will post this, I think I already have posted this. This is its structure at pKa. You can see the ATP molecule, hopefully right here. You see those two purple spheres, those are magnesiums, you're not surprised about that. And this orange-shaped thing that's sitting right in the mouth, right in the cleft, that's that pseudo-substrate region. Okay, let's go back to this. And I've given you that as well in this handout. So again, you can see, here's the M lobe in purple, the C lobe in green. You can see the ATP molecule, two magnesium ions, and that pseudo-substrate here in orange. Now, there are a couple of key features that I want to point out here. First, I want to point out that in order to bind ATP, you can only bind ATP in the kinase domain if it's bound to magnesium. ATP alone doesn't bind very well. You need ATP and magnesium. Now that shouldn't surprise you, because when we talked about how DNA polymerases work and how RNA polymerases work, there were always magnesium ions that were involved in the catalysis. So it shouldn't surprise you that there's going to be magnesium ions involved in this catalytic mechanism as well. Now there's some key features of pKa that I just want to point out that pKa has to do. The first thing it has to do is it has to bind ATP. Okay, and that means in order to bind ATP, if I just remind you what ATP looks like here, I'm just going to draw it this way. There's ATP. In order for this to bind ATP, I got to do two things. First thing that I have to do is I have to bind ATP through some sort of mechanism of regulating the base. So I have to have base recognition. Otherwise, I might mistakenly bind GTP or CTP or TTP or something else. And the second thing that I have to do is I have to be able to accommodate this phosphate, this long polyphosphate chain, this triphosphate chain. And I have to be able to tolerate it after I give up a phosphate to the substrate so that it's only a diphosphate chain. And that is accomplished through another region of the protein, which we're going to call the P loop. P loop for phosphate binding loop. Phosphate binding loop. And in order to do that, in order to accommodate that, that loop is typically glycine rich. And for us, its consensus motif will be glycine, X, XX, glycine. And often, but not always, a basic residue, like a lysine or an arginine. Okay, that's the consensus motif for that P. And again, let's take one more look at pKa. I'd like you to notice a couple of structural features about it. You're going to see some of this again. So first, I'd like you to notice that the N lobe, the N-terminal lobe, is mostly a beta sheet. Can everybody see that? I don't have a laser pointer. Dave, if you would grab me a laser pointer, that'd be great. I don't have one. But you can see, I hope, that N lobe is mostly beta sheet. Got some alpha helices, it's mostly beta sheet. The C lobe is mostly alpha helical. And so I have to be able to recognize those residues. And I have to be able to recognize that P loop. And I'm going to show you in this other handout here, which you also have. This is to basically show you that P loop. So what you see here in yellow, right, that's that floppy P loop that's draping over the polyphosphate chain. Okay? And I've labeled that here as well. So here you can see it's labeled. Here's the gamma phosphate. Here are those two magnesium ions. There's that P loop. Okay? And the other thing that I have to be able to do, I have to be able to do two other things in order for this kinase to work very efficiently. First, I have to, besides binding ATP, I have to bind ATP. I also have to bind the substrate, right? I can't just bind it. I mean, protein kinase A doesn't just phosphorylate every serine, every threonine on every protein. Like, just like chymotrypsin doesn't cut every peptide bond. There's specificity in pKa. And the specificity in pKa corresponds to the substrate consensus motif of arginine, X, where X can be any amino acid, serine or threonine, followed by a hydrophobic residue. I'm going to use the Greek letter phi to indicate a hydrophobic residue. What do I mean by a hydrophobic residue? Leucine, isoleucine, glutamate? Is that a hydrophobic residue? Raise your hand if glutamate's hydrophobic. No, it's negatively charged. Phenylalanine? Yeah, okay. So hydrophobic residues are what's conserved there. What that means is that if I put my eyeball over here and I look into that kinase, so now I'm going to look at the kinase end on like this, and that's where that motif is going to bind, that arginine, RXS hydrophobic motif. That must mean that the kinase has some negatively charged residues over here to bind to the arginines, and it must have a hydrophobic pocket here in order to bind to that hydrophobic motif. Okay? So that's where the specificity comes from. So just like we talked about chymotrypsin, remember we said chymotrypsin had all that catalytic machinery, it had a specificity pocket, which for chymotrypsin was going to be, a pocket that accommodated hydrophobic residues, and for trypsin that cleaves after lysines or arginines, it would be negatively charged. It means that there's substrate specificity here in the pocket as well. But I also have to deal with catalysis. So we've talked about some of the architecture of it. The substrate specificity pocket, the phosphate binding loop. Now the last thing that I want to talk about, and that's what's over here, is the catalytic mechanism. Catalytic mechanism. And I've drawn it on the board, but you don't need to draw it because I've given it to you in a handout. Here's what you have in the handout. Now I'm showing you some of the stuff. No Dave today, huh? Guess not. Okay. What I want to point out here in this handout is the following. First, remember I said that there was going to be base recognition, and I've indicated some of the ways in which that adenine base and the ribose are in the recognizer. I don't want you to memorize the details. Who cares? You can always look it up. But I do want you to realize that proteins that bind ATP recognize the base of ATP. Proteins that bind GTP, which we're going to talk about shortly, recognize the GTP base. They must be able to distinguish them. But here's the guts of the catalytic machine. Okay, so here's the adenine ring, here's the ribose ring, here's where we have our phosphate. So here's our phosphate. Alpha phosphate shown in gray, beta phosphate here, gamma phosphate here. Two magnesium ions. Two magnesium ions shown in red. And here is the substrate that I'm going to phosphorylate. Now I've redrawn the key parts of it over here. So first thing I have to do is I have to position that ATP molecule in exactly the right way in order for me to have effective catalysis. That's catalysis by approximation. So in order to do that, I need to put that polyphosphate chain exactly where I want it. And so I have a bunch of residues that are either interacting with the magnesium ion, and I've drawn the protein residues in orange, the magnesium are these purple spheres. So I have some residues like this asparagine 171 or this aspartate 184, right? That are, or this lysine, which is part of that P loop, GXG, XXXG. Actually, it's not. There's a little stretch, and then that lysine, right? And then that's what's interacting with the phosphate groups to hold them in perfect position. Now I've got my protein. It's got that sequence, right? It's going to have a sequence. It's going to read something like R, R, X, S, hydrophobic. Here's that series. It's going to get phosphorylated. Now, in order to do that, you recall, just like we talked about when we talked about chymotrypsin. Remember in chymotrypsin, that was a serine protease? And in order to make that serine attack the carbonyl, we had to convert that serine hydroxyl into something more like an alkoxide anion. We have to do the exact same trick here. I have to somehow begin to deprotonate this serine to make the serine alkoxide anion. Because what I want to then happen is I want that serine to be able to, that serine alkoxide anion, to then attack the phosphate. And when it attacks the phosphate, then the phosphate will form a bond to the serine and it will break the bond to the remaining ADP molecule, and I will be left then with a phosphorylated serine. And so to do that, this residue, aspartate 166, is going to act as a catalytic base, and it's going to deprotonate that serine. Now when I deprotonate that serine, and I now begin to form my transition state, which I will draw for you. So, I'm going to start out with my protein. I'll try to use the same colors for you. There's my substrate, my serine. That is now going to be attacking. We use two different colors. See it. This is now going to be attacking that. It's going to break. And in order to do that now, I'm going to end up with a transition state that's going to look like this. I'm going to just indicate the substrate by the letter R. That's this whole thing. So I'm going to end up with this. Gee. Why do I feel like I've seen this before? Because that transition state, and I'd like you to notice, what is that transition state? Pentavalent, bipyramidal, metaphosphate. Intermediate or transition state. You've seen this before. You've seen it again and again and again. And I have to stabilize that developing negative charge that I'm seeing here, because as this is forming, I'm beginning to accumulate a lot of negative charge on that phosphate. Okay? That negative charge stabilization happens through two key things. One are the magnesium ions that I've got sitting there, right, which can help disseminate some of that charge. But the other in particular is this lysine residue 168, which is perfectly positioned in order to stabilize the developing negative charge. I'm going to actually stabilize this in developing negative charge in the gamma phosphate. Now, only one more point that I want to leave you with, two more points I want to leave you with regarding pKa. The first is, if we look at the overall structure of pKa, and then of course, I guess I should finish this for you, right? Then of course, what I'm going to be left with is going to be RO plus ADP. So that's my final state. Now, this drawing that I'm showing you here, this shows you the real structure of the two regulatory subunits of pKa in purple and dark blue, and in cyan and green, the two catalytic subunits. And now, let me ask you, suppose I had a substrate that looked a lot like a perfect substrate for pKa. So suppose I had a sequence that read arginine, I can't remember what this is. Instead of serine, I had an alanine, and I followed that by isoleucine. Now that looks pretty much like a good substrate, right, for pKa, except alanine is just a methyl group. Serine, you remember, is a methyl group with an OH group. Would this bind tightly to pKa? Sure it would. It's got almost everything it needs, right, to bind. It matches the consensus sequence. Will it be phosphorylated? No. This is an example of a pseudo-substrate. It can bind, but it cannot be phosphorylated. That exact sequence, RR, whatever, I can't remember, serine, isoleucine, that is the sequence of the regulatory subunit. This red region between the regulatory subunit and the catalytic subunit bind that I showed you in that diagram up there, that is that sequence. So it plugs into the catalytic site, but it can't be phosphorylated. It prevents pKa from being active because it's plugging up the active site of the kinase. Is that part all clear? Okay. Now I want to just say one more thing about, well, I guess I'm going to say a couple more things. Why does this work so well for signal? What is it, if we look at the whole thing, we take that very top panel, epinephrine binds, right, and then it changes GTP on the heterotrimeric G protein, makes cyclic AMP, turns on protein kinase A. What's happening when we look at the big picture? Okay. Well, when the hormone binds to the receptor, okay, now that receptor, can that only activate one GS alpha convert G into GTP? No, because more receptors can come and bind to it and also get exchanged. So one hormone molecule has now suddenly made a whole bunch of GS alpha GTPs. Now each one of those is going to go and bind to adenyl cyclase. Each adenyl cyclase molecule, does adenyl cyclase just take one ATP molecule and make one cyclic AMP molecule? No, it's an enzyme, right? And so each adenyl cyclase molecule is going to make a whole bunch of cyclic AMP. What happens with each cyclic AMP molecule? Well, that's going to activate pKa. I'll put a star to indicate that pKa is active, right? And what's pKa going to do? Is it only going to phosphorylate one protein? No. It's going to phosphorylate a whole bunch of other proteins. So what have I made? I've made an amplifier. So this signaling pathway, where one hormone molecule can exchange GTP for GDP on a whole bunch of GS alpha subunits. And each GS alpha subunit can activate adenyl cyclase to make a whole lot of cyclic AMP. And every 4 cyclic AMP molecules are going to turn on one pKa molecule, but I've made a whole lot more than just 4 cyclic AMPs, so I'm activating a whole lot of pKa molecules. Each pKa molecule then phosphorylates a whole bunch of proteins. So that, and I think I've given this to you in a handout, here's the handout, and I've pointed out to you here all the different amplification steps. Here's an amplification step, here's an amplification step, here's an amplification step, right? And here's an amplification step. And so I've got multiple levels of amplification here. Who's course 6? This is a multi-stage amplifier, right? Maybe you've seen that before. Pretty good being able to make a biochemical amplifier like that. Now, finally, the last point I want to make is what I've shown you in this example is an example of where a 7-transmembrane protein, or a GPCR, was coupled to a G protein that was stimulatory. We called it GS for G stimulatory. And I told you that G stimulatory, right, increases your heart rate, increases your respiratory rate, increases your breathing. But there are also 7-transmembrane receptors that bind to other things, not epinephrine, that can stimulate other types of G proteins. So how about opiates? What happens when someone, like in that movie, overdosed on an opiate, right? She was dead. Her heart rate had dropped. Her respiratory rate had dropped. Her blood pressure had fallen. And so, in fact, there are other 7-transmembrane G protein coupled receptors that couple to other G proteins. And those G proteins have other names. So an inhibitory G protein is called GI. There's another one that we're not going to talk about, but you might see it at some point in the future, and it was in your reading, okay, called GQ. Okay? How does GI work? Why was that, in the movie was that woman nearly dead? Well, she was nearly dead because in this case, I'm showing you what the opioid receptor binds to. When the opioid receptor binds, it actually does the same thing. It stimulates GTP exchange, but G-alpha I, shown here, bound to GTP, interacts with adenyl cyclase, but it blocks it. That's that red arrow. Okay? So the G-alpha subunit still can interact, but instead of stimulating adenyl cyclase, it inhibits it. Levels of cyclic AMP fall, protein kinase A activity goes down, your heart rate goes down, your blood pressure goes down, your respiratory rate goes down. Oh, thank you. Right? Everything goes down. So in this case, I'm inhibiting that. Now what's interesting, and I didn't tell you, but in the case of those G-alpha I proteins, the beta-gamma subunits, which in the case of GS, we think just hang around. Here, the G-beta-gamma subunits can also signal. Okay? And so all parts of this pathway are active, and I can use it to stimulate a process or to inhibit a process. Again, all of this, everything I've shown you, works by allosteric, right? We've gone through that quite a bit. But remember again, those 3 switch regions are what are engaging with adenyl cyclase to drive adenyl cyclase's activity. It's also allosteric. G protein binds to one point on adenyl cyclase, that turns on the catalytic domain somewhere else, the whole thing. You've seen many examples now of allosteric. Questions about G protein coupled receptors. I'm going to change, I want to talk about one other really important group of receptors which work similarly but differently. So now I want to talk about receptor tyrosine kinase. Receptor tyrosine kinase. And I want to contrast them with GPCRs. So let's first talk about the architecture of the signaling network. Okay. So just to remind you, a GPCR, this was our 7-transmembrane G protein coupled receptor, acted through a G protein, and then that acted in some way on a kinase. Receptor tyrosine kinases, which I'm from now on going to abbreviate as RTKs, for receptor tyrosine kinases, they work a little differently. RTKs contain a kinase as part of their structure. So the kinase comes first, and then that, among other things, acts on G proteins in an indirect way. Different G proteins, not heterotrimeric G proteins, small G proteins, and then those G proteins can do a wide variety of things, including activating other kinases. So that's a fundamental difference. Now I'm going to put up the handout that I gave you. We're going to walk through this one step at a time. Now, let's talk a little bit about what type of things are receptor tyrosine kinases. RTK ligands. By ligand, I mean something that's going to bind to the receptor tyrosine kinase. Now, you know, I told you that for GPCRs, G protein coupled receptors, those ligands are things like small molecules like epinephrine or other small hormones or growth hormone. It can be, opiates. It can be neosynephrine. It can be odorant molecules that you smell, right? It can even, believe it or not, it can actually work through photons, through a rhodopsin in your eye. But in this case, RTK ligands in general tend themselves to be proteins. And the proteins that these ligands are, in general, are growth factors. And these include things like epidermal growth factor, which I will abbreviate as EGF, fibroblast growth factor, which I will abbreviate as FGF. All of those growth factors typically work through this process, and that's because one of the main things that these G proteins do, besides activating kinases, is they drive overall cell proliferation. Now, most of these are, and I'm going to add one more thing that's a growth factor. Maybe you hadn't thought of it as a growth factor, but that would be insulin. Insulin is another growth factor that works through binding to a receptor tyrosine kinase. And for the most part, not all of these, but for the most part, most of these receptor tyrosine kinases start out as monomers. And I'm going to draw them like this. They have a part that's outside the cell. This is the outside. This is the inside of the cell. And this is where they're going to bind their growth factor. They have a transmembrane segment. They don't have 7 transmembrane segments. They only have 1. And then they've got this cytoplasmic tail that has a kinase as part of it. There's something unique about this. Okay, so I've got 2 of these, let's say, sitting in the membrane. That's my terrible art skills. And they have this long cytoplasmic tail sitting like this. Okay. Now, what's unique about this kinase that these molecules contain is that this isn't just any old kinase. This in particular is a tyrosine kinase. What I mean by that is it preferentially phosphorylates a tyrosine residue. And it's going to turn that tyrosine residue, I guess I should draw for you in case you've forgotten. This is the side chain of tyrosine residue, and it's going to now turn that tyrosine residue into a phosphotyrosine. phosphotyrosine, which I'm going to abbreviate. I may abbreviate this as PY, phosphotyrosine. That's what's unique about these receptors. What happens is the following. When the ligand binds to the receptor, this is all in your handout. When the ligand binds, now I have two receptors that are dimerized with each other. And these receptors cross-phosphorylate each other. And they specifically phosphorylate tyrosine residues that are sitting in their C-terminal tails. So that when the ligand is bound, and I'm going to indicate that in red color, now those tyrosines become phosphotyrosine. That phosphotyrosine, I'm going to have to use the other board to draw the next part of this. Through some process that we're going to, so leave some space here and here. Through some process that we're going to explain a little bit more of in a moment, are able to turn a G protein from its off state into its on state. I need a couple colors of chalk. Now I'm going to be able to take some kind of a G protein. I'll talk about this in a moment. A small G protein, which is bound to GDP. And I'm now going to convert that G protein. This GTP is going to come in. That GDP molecule is going to fall off, and I'm going to be left then with my molecule in an alternative form because it is now bound to GTP. And in this state, this molecule is now active, and it is going to drive, among many other things, cell proliferation. If you're writing this in your notes, I want you to leave some space over here because we're going to talk about shutting the process off in a few minutes. What I want to talk about next is how do I go from phosphorylating these tyrosine residues to being able to turn on this small G protein. And then we'll talk later about what that small G protein is. So it turns out I need to couple this, as you can imagine, to some kind of a GTP exchange factor. Right? And the factor that I'm going to couple this to is a GTP exchange molecule called SOS, S-O-S. This is my GTP exchange factor. So somehow, binding of a ligand to a receptor tyrosine kinase has to turn on the GTP exchange factor. And that GTP exchange factor is going to cause this G protein that's bound to GDP to convert to the GTP bound form and do that. The reason it's called SOS, again, you can blame it all on the geneticists. You know, geneticists name things after phenotypes. They don't name things like it would make biochemical sense. So there was a fly mutant that was missing a photoreceptor segment called 7, and this mutant recapitulated that, so they called it son of 7less, or SOS. Geneticists. What can I tell you? Okay? but that's a GTP exchange factor. How do I couple, how do I link activation of the receptor and its own phosphorylation on tyrosines to turn it on a GTP exchange factor? And the way I'm going to do this is I'm going to use a really, really clever approach. Okay? I'm going to draw it all happening here just because it's just going to be a little easier to do. I'm going to have to use a molecule that's going to, so I'm just going to draw SAS over here for just a moment. This is the same SAS molecule. Okay? SAS. I don't want you to think it moves, I'm just drawing it over here. That's the same as drawing it over there. Okay? How do I couple SAS, right, to that tyrosine phosphorylation? And I have to do that by using an adapter protein. I want to link SAS to this autophosphorylation event. That means I have to do two things. Okay? I'm going to have to construct a molecule here that is capable. I'm going to draw it a certain way of recognizing that at least one site on this receptor has been phosphorylated, and it's linked to this molecule SAS. And the way I'm going to do this is I'm going to use modular protein domains. Okay? I'm going to use protein modules to do this. Protein modules. Or protein domains. Now we talked a little bit about domains, and I've already snuck in on you the idea of a domain when I talked about kinase domains, and I talked in PKA about that kinase, that catalytic subunit as being a domain. What do I mean by a domain? I mean a part of a protein that folds independently and it has some kind of autonomous function. So meaning, if we think of proteins in general as beads on a string, here's my protein. These beads on a string might all be domains. Now, in order for allostery to work, what I might want is I might want those domains to interact with one another. Right? So here's one domain, there's another domain, there's a third domain. I might want those domains to somehow interact with each other, right? Because that's going to give me the ability to use allostery, right? Where binding of something to one domain might affect its interaction with another, but that's the idea here. And this molecule that I've drawn in blue that is going to connect for us this phosphorylated growth factor to this GTP exchange factor is an example of one of those modular protein domains. This molecule is called growth factor receptor binder 2, or GRB2, GRAB2. Growth factor receptor binder 2. Okay? And it has two domains. It has a domain called an SH2 domain, and it has two domains called SH3 domains. And I've given this to you in the handout, and I'm just going to tell you examples of what these are examples of particular domains. So in this case, examples, SH2 domains specifically recognize phosphotyrosine residues. So that after I phosphorylate the tyrosines, these SH2 domains can bind. Now, I've given you this handout. This is an example of an SH2 domain. It happens to come from a different molecule. You can see the SH2 domain is mostly beta strands, 2 alpha helices. And shown in here in stick is that phosphotyrosine. And you can see that phosphotyrosine is specifically bound to part of that SH2 domain. So that SH2 domain can detect that the growth factor receptor has been activated because it can bind to that phosphotyrosine. SH3 domains, these are things that bind to polyproline sequences. And an example of that is found on SAS. SAS has polyproline regions. And so these two SH3 domains bind to SAS. And that's what's in the handout that you have here, where I'm showing you. Here's the monomeric receptors, they dimerize, they phosphorylate. Here's that growth factor receptor binder 2. It uses its SH2 domain to bind to the growth factor receptor. It uses the SH3 domains to bind to SAS, and that then stimulates the GTP exchange of RAS. Now, what is that small G? You know, it's 10:20. Why don't we take a 1 minute break? I can't tell you much about it, except I just told you it was going to be a small G protein. Now, the name of that small G protein, I suspect, is something you've heard before. Who's heard of RAS before? Raise your hand. RAS is the small G protein. It is downstream of these receptor tyrosine kinases. Now, why is it called RAS? Anybody know why it's called RAS? Because it was first identified in a connective tissue tumor in rats. That connective tissue tumor is called a sarcoma. And so RAS stood for rat sarcoma. Okay? That's where the name comes from. But many people don't know that. But it came from a gene that was found in rat sarcomas. That's why it's called RAS. Now, importantly, okay, this is different than those heterotrimeric G proteins we talked about. It's not like GS, it's not like GI, it's not like GQ. It's different because first it's small. Its molecular weight is only about 21 kilodaltons, right? Pretty small. Remember I told you the GS, the alpha subunit was 45 kilodaltons, and the beta subunit was 35, and the gamma subunit was 7. This is only 21 kilodaltons. And in fact, if you look at what RAS looks like, I'll show you a picture of it. You have it in the handout. I'll show it to you. This is what RAS looks like. Boy, it's got a sort of central beta sheet and a couple alpha helices. Doesn't that look a little bit like that N-terminal lobe of the protein kinase? Remember, I showed you that N-terminal lobe, right? And I said to you, look, this is what binds ATP. It's got a central beta sheet and then, a couple of helices. This looks very similar. The only difference is where the N-terminal lobe of protein kinases bind ATP, this small molecule binds GTP, right? It binds GTP, and then it undergoes a conformational change. And then it will eventually hydrolyze the GTP to GDP, and that will again cause another conformational change. Now, you can bet that in order for this to bind GTP, going to use a bunch of the same tricks that you've already seen other molecules use. So, how is it going to bind? Why is it going to bind to GTP and not to ATP? Same trick, same chemical trick you've seen before. It's going to use a base binding region, okay, that has specific base recognition. Unlike what I drew for you up there, where the base recognition was for an A, now the base recognition is going to be for a G. The second thing it's going to use, is it's going to have a way to recognize this phosphate group. So it is also going to use a P loop. And guess what the sequence of that P loop is going to be? GXG, XXGK. Exactly what you've seen before. And furthermore, it's only going to bind magnesium. Sorry, it's only going to bind the GTP in the presence of magnesium ions. Wow, this is so boring. Polyphosphate, magnesium. Can you guess what the transition state's going to be? Yes, we're going to get there. Okay. That's RAS. Again, here's what RAS looks like in the GTP bound state, a critical magnesium ion that is required. It's got that central beta sheet we talked about. Now, there are two regions of RAS. Remember when we talked about heterotrimeric G proteins? This is just to show you. Here's that P loop in red. See that red P loop draping over the phosphates? Here's the guanine ring. Here's the ribose ring. Here's the phosphates. Here's that magnesium. Here's that P loop. Here's the GXG, XXGK. There's that lysine, which just like in that diagram up here, was helping to stabilize, right? Helping to hold the magnesium in place and stabilize the fluid state. It's going to be the same thing. Now remember I told you that there were two regions remember we talked about heterotrimeric G proteins? And I said GS alpha changes its shape when GTP is bound compared to GDP, and I said that there were really three regions in those GS proteins that change their shape, and we call them switch regions. So RAS and every other small GTP binding protein that we're ever going to talk about also has two switch regions, right? And those switch regions are called switch 1 and switch 2. And I'm going to show you in this movie, and I will post this parallel. I'm going to show you the change. GTP, so first look at the red and green. GTP, GDP. GTP, GDP. Now look at the loops. Loops are switching. I'm going to show it to you again. See how the shape of these switch regions changes? Now it's subtle, right? You see switch 2 appears to change a bit more than switch 1, right? But those two switch regions are changing. And when they change their shape in the GTP bound state, that's when RAS is able to signal. And in the GDP bound state, it's not able to signal. Let's talk a little bit about the catalytic mechanism. So you can imagine what the catalytic mechanism is. Catalytic mechanism. The catalytic mechanism has to be this. I start out with GDP, O, P. And that phosphate starts out in a tetrahedral state. Now, a water molecule is going to come in. The water molecule is going to come in. And if I activate that water molecule appropriately, that water molecule can attack that phosphate group, and I can begin to break this bond with the GDP. And so I will end up in a transition state that's going to look something like this. GDP, O, P. I'm not going to write it. What is the transition state? Boring. Biochemistry seems to have found one reaction that it likes, and it just keeps doing it and doing it. Okay. Now we can actually see that transition state. We can capture that transition state in detail, and we can do it because we can crystallize RAS. But instead of, that's going to be hard to crystallize RAS with GTP, right? RAS is going to hydrolyze GTP. Slowly, I admit, but it's going to hydrolyze GTP. But in place of this transition state, I can use GDP, and I can use aluminum fluoride, one of two forms of aluminum fluoride. Because now if everything works well, I'll be able to crystallize RAS or whatever I want with a nucleotide diphosphate, an aluminum fluoride molecule, and a water molecule, and that will resemble this transition state. So in fact, that was done. This is the crystal structure. not RAS. This is the structure of a related protein. Okay, so this is I'm going to show you in detail. This is the structure of a related protein. It's essentially RAS. Here's the GDP. Here's what we do the gamma phosphate mimic of aluminum fluoride. And here is a water molecule. And I'd like you to notice what's happening here. What's helping to activate? What's helping to activate this molecule? So there are a couple residues that play a really critical role. So key residues in RAS. So if you look at it, you can see, well, it looks like glutamine 61 and threonine 35 play a really important role, right? Because these two residues seem to be orienting the water molecule in a perfect position so that it can attack that gamma phosphate. Okay? Everybody still with me on that? Now, in addition though, in addition to helping activate that water molecule, there are also residues in that P loop of RAS. I'm going to put this, let me see if I've got this. So here I'm showing you RAS bound to GTP. This is in your handout. And I'm focusing here on that P loop. Here's that GXGX. It turns out in this case it's GXGXGK. So, and here's the G15, here's K16. You see that lysine residue. That lysine residue is playing a pretty important role here in coordinating the magnesium and stabilizing as well the additional phosphate groups in GTP. Now, there's one problem though, and I have to tell you about it, and that is that compared to heterotrimeric G proteins, remember I told you heterotrimeric G proteins could turn themselves off, right? They would do the 7-transmembrane receptor, puts GTP into the 7-transmembrane into the heterotrimeric G protein, and the G protein then auto-hydrolyzes that GTP after a short period of time, the GDP to turn the process off. Okay? The important point I want to tell you is that the intrinsic GTPase rate of RAS is slow. It will hydrolyze GTP, but it does so relatively slowly. And so to help it go faster, in the case of these molecules, there are molecules that facilitate this, and these molecules are called GAPs, GTPase activated proteins. Or GTPase activator proteins, GAPs. And so, going back to this original diagram, in order for me to turn off RAS GTP, now I have to have a water molecule come in, and ideally a GTPase activator protein, and that will lead to a phosphate group falling off, okay? And will convert RAS from its GTP bound form to the GDP bound form. Now, for exams, I'd like you to think about if I use radioactive GTP and I add a gap or not, will the GDP be radioactive? If I use radioactive GTP and then I incubate it with a gap, is the GDP here going to be radioactive? Now look, I've got radioactive here. I'm going to radio label the G, the base, so the base has the radioactivity. Is this going to be radioactive or not? Who says it is? Yeah, you're right. Of course it is. Now, in contrast, at the very beginning of the process, way up there, okay, when SAS is coming in, okay, if I start off with RAS, GDP, that is radio labeled, and now I add SAS and I get the exchange, is this going to be radio labeled? Is there going to be radio labeled here? How many people say yes? No, you're right, because it's going to fall off. So you can expect on an exam, I might give you different radio labeled stuff and ask you where it's going to end up. Okay. Now how is it? How is it that the intrinsic rate, if the intrinsic GTPase rate of RAS is so slow, how is it then that these gaps are able to enhance it? Well, remember, the rate limiting step in any enzyme is stabilizing what state? The transition state. I have to stabilize the transition state. I have to stabilize that developing negative charge on the phosphates. And so this is an example of one of those GTP activator proteins. This comes from my friend from Smurden. This, I'm going to show you, this isn't RAS, it's a homolog of RAS. What would I call a homolog of RAS? Why don't I call it RAS homolog, R-H-O. That's rho. Okay? That's another small GTP molecule. There are a whole bunch of them. RAND, rho, RAS, RAL, RAP, there are a whole bunch. They're all RAS homologs. Here you see RAS. Here you see, or rho, bound to that GDP aluminum fluoride. Think of it as GTP. And here it is bound to that GTPase activator protein. What amino acid is that? I'll show it to you in a little more detail. That amino acid that the gap is inserting in. What amino acid is that? It's an arginine, okay? The gaps at work because they insert an arginine finger into the catalytic cleft in order to stabilize the transition state. I'm going to write that down. Gaps use an arginine finger mechanism. To stabilize the GTPase transition state. That's how a gap works. Now, why do you think heterotrimeric G proteins? I said that they have a pretty good intrinsic GTPase structure. What do you think is different about a heterotrimeric G protein compared to these small G proteins? What do heterotrimeric G proteins have that small G proteins require a gap? The arginine finger. Okay, a heterotrimeric G protein has its own arginine finger. It can stimulate its own autocatalysis in a way that a small G protein can't. A small G protein requires a gap. A heterotrimeric G protein uses the same arginine finger mechanism, except it has its own arginine finger. It is capable of stimulating autocatalysis. Now, let me ask you, what would happen if RAS got stuck in this GTP bound form? What if RAS couldn't hydrolyze that GTP anymore, right? It would stay stuck in that one state. What signal would that be saying? That would be saying proliferate, proliferate, proliferate. Guess what? Many, this is in your handout as well, many oncogenic mutations in RAS involve residues involved in GTP hydrolysis. So RAS mutants, particularly glycine 12, that's part of that P loop, threonine 35, and glutamine 61, right? Yeah. Glutamine 61 are common mutations. Why? Because I showed you that glycine-rich loop drapes over the phosphates. You need that to catalyze to catalyze exchange of GTP for GTP. Or these threonines and glutamines that are responsible for coordinating the water molecule, these mutations cause cancer. And now you know why. Okay, we do have time to do sketches. Let's do sketch of plots very quickly. Now, suppose I I wanted to actually measure the affinity of RAS for GTP. Okay? Or I wanted to measure the affinity of protein kinase A's regulatory subunits for cyclic A and B. I need a way to quantify this mathematically. So let's talk a little bit about radioactivity and RAS. So what I want to do is I'm going to ask the question, I want to quantify RAS binding to GTP. And the way I'm going to do the experiment is the following. I'm going to take in my test tube a solution, it's going to have some buffer in it, and it's going to have RAS. Okay. And now I'm going to add to that some GTP is radioactive. This asterisk means radioactive. Okay. And I'm going to incubate these together. And I'm going to wait a certain amount of time. And then I'm going to precipitate the RAS with trichloroacetic acid. Trichloroacetic acid. You've seen that before. When we talked way back at the beginning of the course about how they purified DNA polymerase, you learned that they did that because they incubated this DNA polymerase with a radioactive substrate, and then they precipitated it with trichloroacetic acid, and then they put it on a filter. Okay, so now I've got a filter sitting like this, and I'm going to pour this stuff through it, and the protein, the RAS protein, is going to get stuck on the filter because it's been precipitated, and if it's bound to the radioactive GTP, then I'm going to have GTP on the filter. And so I'm going to count the amount of radioactivity I get. Count radioactivity. And I'm going to make a plot here of the amount of radioactivity in the precipitate, that is the amount of radioactive GTP in the precipitate as a function of GTP concentration. And when I do the experiment, what I find is I get a curve that looks like this. I get some maximal amount of binding, right? Radioactivity in the precipitate equals RAS binding. And I find some maximal amount of binding. We'll call that Bmax. Okay? Now, you've seen the shape of that before. What's that shape called? Rectangular hyperbola. It's just like oxygen binding to hemoglobin. Okay. Now, you know that in Yaffe later, you can only say something science if you can push the electrons around and describe it mathematically. So let's see if we can describe it mathematically for it. Okay. So here's the formula, the way we're going to use this. R is the receptor in general. L is the ligand. We can say that our receptor has some Kd, which is equal to receptor ligand or the receptor ligand complex. Okay? And we're going to say that the occupancy of the receptor, occupancy, we're going to give this some level, y. Sometimes it's called theta. I'm just going to write theta here because if you take higher courses in course 20, you might see it written as theta. And that's simply equal to the fraction of the receptor that's bound to ligand over the fraction of the receptor over the total amount of receptor. And this is, of course, simply equal to, I'm just going to rewrite it up here. This is, of course, just equal to, I'm going to write y equals the amount of bound receptor over the total amount, which is the amount of free receptor plus the amount of bound receptor. Yeah. I'm going to substitute in for this. I'm going to substitute in for receptor, for R. I'm just going to substitute in R here as Kd times RL over L. All I'm doing is substituting from this equation. And now what I'm going to find is that Y is equal to the concentration of ligand over KD plus the concentration of ligand. Now that should be a big relief. You've seen that before. This is the exact equation for myoglobin binding, right? This is just myoglobin binding all over again. Except instead of P50, it's the KD, and instead of the PO2, it's the concentration of ligand. And that equation gives us exactly the rectangular hyperbola that you saw here. Okay? Now, how do we come up with this? What I really want to know is I want to know how much receptor do I have, right? What's the amount of receptor that I can bind to? Now, you might say to me, oh, this is easy. I really want to get the KD first, right? How tight does it bind? How much receptor do I have? Now, you might say to me, oh, Professor Yaffe, that's easy. I'll just fit this curve, right, to a rectangular hyperbola. Yes, you can do that, right? That's what we do in 2025. But remember, in the old days, before computers, the name of the game was linearize nonlinear equations, and so that's what we're going to do. We're going to try to linearize the equation for KD. Okay? So we're going to start out then again with our KD equation, and what I'm going to do to start with is I'm going to take this KD equation and I'm going to multiply it by RL on both sides. Okay? So what I'm going to write then is that the KD is equal to, in place of free receptor, I'm going to write the total amount of receptor minus the amount of receptor that's already in the RL bound state times the concentration of ligand divided by RL. I'm going to multiply in this case now. I'm going to multiply by RL on both sides to clear this, and now I'm going to have KD times the concentration of RL is equal to this top part. The concentration of receptor total minus the concentration of RL, concentration of ligand. Now I'm going to divide everything. And when I divide both sides by that, what I'm going to end up with then is this. RL over L is equal to R total over KD minus RL. Let's think for a minute about what that equation tells us. What that equation tells us is that this term here is the bound ligand over free ligand is equal to some constant. Okay, because the R total, well, there's some total amount of receptor in my assay, and KB is a fixed number. It's equal to some constant, right? Minus the concentration minus the amount of bound ligand over KB. And so now I'm going to make a new plot. Now what I'm going to do is I'm going to make a plot, and I'm going to plot for ligand bound over free. That's RL over free. On the y-axis. So that's this, bound over free. That's ligand bound to the receptor divided by free receptor. And here, I'm going to plot that against the amount of bound ligand. So that's RL on the x-axis. When I do that, I'm going to get a bunch of points. The slope of this line is equal to -1 over KP, because I've just plotted bound ligand over free ligand. versus bound ligand. The slope of that is minus 1 over Kd. That's Kd. That's how I'm going to get Kd. Now what about this intercept? At some point, I'm going to have here at some point the x-intercept. And I want to know what that x-intercept is. Okay? That x-intercept, this occurs, the x-intercept occurs when the concentration of ligand goes to infinity. Why? Because when the concentration of ligand goes to infinity, this first term is concentration of bound ligand over infinity. That's going to go to 0. The only way that that can happen, at that point, when the ligand goes to that point, this occurs when the concentration of bound ligand is equal to the concentration of receptor total. Okay? And I can derive that for you by just taking that first equation. Because that first equation is 0, right? When RL, at this point, at the x-intercept, when RL over L goes to 0, that must mean that R total over Kd has to equal RL over Kd. Okay? And so this point tells me, this amount tells me the total amount of receptors. So from this one curve, which has a name, it's called a scatter plot. Where I'm plotting bound over free ligand versus bound ligand. The slope tells me the Kd, and the intercept tells me the total amount of receptors. I want to ask you 2 questions. Okay? If I had more than one binding site, the receptor total could be more than the number of receptors. If I did this for the regulatory subunit of pKa, I would find the receptor total was twice the actual amount, each subunit bound. The second thing I want you to think about over the break is, what if I had cooperative binding, where the first binding site made the second binding site tighter or weaker? Would I get a straight line, or would my line have a break in it, or a different shape? The last point I want to make, very last point I want to make today, before I let you go, is this. You probably wonder, why are there all these ATP binding molecules and GTP binding molecules? Right, and in fact, we're just starting. After the break, you're going to see a whole bunch more things that are linked to nucleotides, right? You're going to see NAD, which is basically this looks like ADP linked to nicotinamide. You're going to see FADH2, that's another ADP molecule, right? Which is linked then to this other weird thing, flavin-adenine dihedral. And we talked, and actually, the best one of all, my favorite one, is this molecule here, which is called coenzyme A, or CoA. You've heard of CoA before. Okay? Here's CoA. It's again an adenine, it's again basically ADP, linked to a vitamin, linked of all things to a thiol, a chunk of cystine. Why in the world would nature ever make something like that? What animal is this? This is a duck-billed plasmid. This is the weirdest animal I think you will ever see. I love these animals. Fly sleep with them. Okay? So this is my stuff now. So why do I say it's the weirdest animal? Because it's got a stinger that produces venom like a reptile. It's got a beak like a bird that lays eggs, but then it breastfeeds like a mammal. Can you imagine a weirder thing? It's like you took parts of 3 different species and glued them together. What does that have to do with acetyl-CoA? Well, nature had figured out, this is now I'm going to teach you molecular evolution simplified. Okay? Nature had figured out how to use nucleotides. It was using them to make RNA. It was using them to make, DNA. Okay? And now suddenly it wanted to fly. It wanted to do, it wanted to do all kinds of other stuff besides just make DNA and RNA. Now, if I asked you to build a plane, and you were here in Aerolastro, you would probably build something like this, okay? But nature doesn't work that way. Nature says, look, I got a car, I want to fly, here's what I'm going to do. I'm going to paste some wings onto the car I've already got, okay? That is acetyl-CoA, or that is CoA. CoA, and all those other molecules that you're going to learn about after the break, is this. It's nature saying, look, I know how to recognize bases. I know how to recognize ATP or GTP or whatever. I'm just going to slap anything else I need onto ATP or GTP. That's why we charge amino acids with ATP to make the aggregated amino acid, right? The activated one. That's the secret of molecular evolution. Take what you got, make it work. This is the last lecture I will give to you. It's been a real honor teaching you. I hope all of you go on to become biochemists. Thank you.